Pollination and double fertilization in plants are followed by embryogenesis and endosperm development. These processes in reproductive growth represent the final stages of the life cycle of plants. Essentially, these latter stages will produce seeds that will repeat the same cycle of vegetative and reproductive growth and ensure the perpetuation of the species. In this lecture, we will examine how the fertilized egg cell develops into an embryo in dicot plant species. From previous discussions, you already know that double fertilization produces a zygote and a primary endosperm. The zygote is a diploid or has two copies of the full chromosome complement of the species, while the primary endosperm is a triploid or has three copies of the full chromosome complement of the plant species. Both the zygote and the primary endosperm will undergo several rounds of cell division to develop into a mature embryo and endosperm respectively. The mature embryo and the endosperm, along with the seed coat, will make up the primary parts of the seed. In this section, we will look at the detailed process of how a zygote resulting from the fusion between the egg and a sperm cell develop into an embryo. The process of embryo development or embryogenesis can be divided into three stages. The first stage is morphogenesis, followed by embryo maturation, and then finally desiccation. Morphogenesis is the phase when the polar axis, meaning the shoot and root of the plant body, is defined. This is also when the embryonic tissue and organs are formed. Following morphogenesis is embryo maturation, which is characterized by the accumulation of storage reserves. Finally, the embryo undergoes a controlled process of desiccation or drying and enters a period of developmental arrest. Let's now take a closer look at what's happening at the cellular level during these different stages, starting from morphogenesis. In the succeeding slides, the morphogenic events that we will talk about are characteristic of a dicot plant. The zygote, which is the product of the fusion between the egg and the sperm cell, undergoes an asymmetric cell division. This division produces an apical and a basal cell. The apical cell is the smaller cell on top, and the basal cell is the relatively bigger cell at the bottom. These two cell stage signals the start of embryogenesis. The asymmetric cell division provides polarity to the embryo. Plant polarity essentially determines which parts are going to become shoots and which parts will be the roots. The smaller apical cell on the top of the basal cell contains most of the cytoplasm from the original zygote. The apical cell will give rise to the hypocotyl or the embryonic stem, as well as the shoot apical meristem and the cotyledons. The bigger basal cell will give rise to the hypophysis and also to the suspensor. We will define these two structures in the succeeding slides. Now within the ovule, the zygote will be located near the micropyle, which is the small opening where the pollen tube goes through during fertilization. At the two-cell stage, the embryo will be oriented like so, just like what you see here, with the basal end oriented more outwards the micropyle. The two-cell embryo then undergoes 2 rounds of longitudinal division and 1 round of transverse division, resulting in an 8-cell embryo. During the 8-cell embryo stage, 4 domains are going to be very obvious. What are these 4 domains? We have the apical embryo domain, we have the central embryo domain, we have the basal embryo domain, and then we have the suspensor. The apical embryo domain will give rise to the shoot apical meristem. The central embryo domain will give rise to the hypocotyl, root apical meristem, and parts of the cotyledon. Collectively, the apical and the central embryo domain contribute to the embryo proper. Now, the basal embryo domain contains the hypophysis. The hypophysis is the structure that will give rise to the radicle or the embryonic root, as well as the root cap. Lastly, we have the suspensor. The suspensor is the structure that connects or attaches the embryo to the endosperm for nutritional purposes. The 8-cell embryo then undergoes a periclinal division to produce a 16-cell embryo. A periclinal division is a division parallel to the embryo surface. During this stage, the 4 domains get more defined. What are those 4 domains again? We have the apical, central, and basal embryo domains, as well as the suspensor. Aside from these domains getting more defined, the first recognizable tissue is formed, and this tissue is called the protoderm. The protoderm is the precursor of the epidermis, and in this diagram, that is a set of tissue that is encircled in red. Further division of the 16-cell embryo leads to an embryo at the globular stage. At this stage, three basic tissue systems can be observed: the dermal, ground, and procambium tissues. The dermal tissue originated from the protoderm and constitutes the epidermis. The ground meristem will give rise to the ground tissues, including the pith and cortex. Lastly, the procambium will give rise to the vascular tissues, including the xylem and the phloem. Following the globular stage is the heart-shaped embryo. As the globular embryo undergoes division, the cotyledons or the embryonic leaves begin to form. The early development of the cotyledons give the embryo a heart-shaped appearance. In monocots, only a single cotyledon forms. The continuous growth of cotyledons and axis elongation of the heart-shaped embryo results in an embryo at the torpedo stage. The upright position of the cotyledons gives the embryo its torpedo shape. Aside from the characteristic shape of the embryo, there are two events that define the torpedo stage of embryogenesis. The first one is the programmed cell death that leads to the degeneration of parts of the suspensor complex. As I mentioned earlier, the suspensor is a supporting structure that connects the embryo to the surrounding maternal and endosperm tissues. By attaching the embryo to the endosperm, the suspensor is able to facilitate nutrient transfer. It also pushes the embryo proper to the embryo cavity as it develops. During the torpedo stage of embryogenesis, the embryo pretty much utilized all the nutrition from the endosperm for its development. Since there is no longer a point in having the embryo connected to the endosperm, the suspensor will start its degeneration. The second event is the establishment of the shoot and root apical meristems. In this illustration in the left, we have the shoot apical meristem here and the root apical meristem in this region of the embryo. Lastly, we have the embryo at the mature stage. The growth of the torpedo stage embryo, which is accompanied by the total disintegration of the suspensor complex, leads to the maturation of the embryo. The disintegration of the suspensor complex signals the end of embryogenesis. When this happens, the mature embryo will start post-embryonic development, where it will undergo a controlled process of desiccation or drying. Just like during pollen maturation, the dehydration process is meant to temporarily arrest or stop any metabolic processes that puts the embryo in a state of dormancy. It will remain dormant until specific requirements for germination are met. This slide summarizes the different morphogenic events in dicots as it progresses from a 2-cell stage to a 16-cell stage to the heart-shaped stage, the torpedo stage, and to the mature embryo stage. I want to reiterate here that the asymmetrical division that started from the zygote is very important in establishing the polarity of plants. In this slide, you can see the cells originating from the apical cell and the basal cell. Those that originated from the apical cell are colored yellow-green and now form part of the shoot. The cells that originated from the basal cell are colored pink and now form part of the root cap and also the radicle.